All right, everyone, let's get started. Welcome everyone to our next topic in our Refsum webinar series. Today, we're here to talk about gene therapy as a potential future therapy for Refsum disease. I'm Christy DeMarco, and I'm the founder and president of Global DARE Foundation, and I'll be your host today. We are honored to have Ryan Butler, Ph.D., who is the assistant professor at UT Southwestern Medical Center as our presenter today. Let's review the agenda. Let's see. Oops, here's our beautiful pictures. All right, so our agenda, I'm gonna go a little over our Global DARE Foundation's mission. I'm gonna hand it over to Ryan, who will present on gene therapy. And then we'll move into a question and answer session where you'll have an opportunity to ask questions of Ryan about his presentation. So first up is to talk a little bit about the housekeeping details. All the participants are in listen-only mode. There'll be a few ways that you'll be able to ask your questions. Participants following in Zoom can type their questions in the Q&A box. You'll see that on your toolbar. You can also raise your hand during the presentation, either through the toolbar or by pressing Alt-Y. And then if you're dialing in, you can press *9 on your phone, and that will raise your hand, and we'll be able to see that and be able to unmute you so that you can ask your question of Ryan. After the presentation, I'll be moderating the questions, and I'll start with the Q&A box in Zoom, and then I'll move into the dial-in participants and then the others that have raised their hand online. Today's session will be recorded for later viewing on our Global DARE Foundation website. All right, let's talk a little bit about DARE's mission. So Global DARE Foundation was established in October 2019. DARE stands for Defeat Adult Refsum Everywhere. Our mission is to promote worldwide awareness and better quality of life for those who are diagnosed with adult Refsum disease. Our goal is to support research, education initiatives, awareness campaigns, and advocacy. Driving research is really at the center of what we do because we dare to believe that there's a cure for adult Refsum disease. Before I pass it over to Ryan, I do want to make you aware of an upcoming webinar, which really connects very well into what Ryan's going to be talking about, because to support research, having data on the progression of a disease is so important, and we are going live with a patient registry for adult Refsum disease. It'll be the first ever patient registry for this disease, and us as patients can contribute to research by participating in this patient registry. So we'll have a webinar on Monday at noon Eastern time where myself and Sanford Corge, which is the registry platform that we'll be using, will provide an overview of the system and the questions and provide a demonstration of that system. So, and then you'll be at that point be able to register within the platform. So now I'll hand it over to Ryan, who will give us a great presentation on gene therapy. Over to you, Ryan.
Thank you. Am I on mute still?
No, you're all set.
Okay. Good morning, everybody. Early morning, as is the case here. Today, I will be talking about gene therapy for adult Refsum disease. Probably more relevant is that this is the prospects of using gene therapy for adult Refsum disease. I am an assistant professor at UT Southwestern Medical Center. I've been working on gene therapies for about 2 years now. Okay, next slide. So the promise of gene therapy is to fix the genetic disease at the source. If you fix the DNA, you solve the problem permanently. Sorry, I'm just moving our videos real quick. So there's a book that came out a couple years ago called The Forever Fix. It's about the story about a boy who had a hereditary blindness and was essentially cured using gene therapy. This story also goes through all the trials and tribulations of the field of gene therapy and how we got to where we are right now. Next slide. So gene therapy conceptually began in the early 1970s. There was a theoretical science paper released by Friedman and Robin in 1972, which basically describes the possibility of using the innate features of viruses to correct genetic defects. It's essentially comprised of an engineered viral capsid in a DNA construct. And so it works but as gene replacement therapy. Essentially, you are transducing a functioning copy of a gene to overcome an improperly functioning gene. It could also be used to manipulate existing gene regulatory systems. Next slide. So as of December of last year, there have been over 3,000 clinical trials approved, 250 of which use adeno-associated virus. And importantly for what I'm talking about today, at least 27 approved clinical trials. It's actually probably a lot more than that now, but at least 27 approved clinical trials have been made for a liver-directed gene therapy. Next slide. So how does gene therapy work? This is the simplified version of this, but basically you have to identify the defective gene in patients, and then you package a working copy of the gene into an engineered virus and then deliver that virus back into the patient. Okay, next slide. And so I'm going to just start off with talking about some of these success stories. And this first one was a study which was published back in 2012. It came out of China about a child with L-amino decarboxylase deficiency. And this was the first person to actually receive an experimental gene therapy vector directly into the brain. So go ahead and Christine, play the left video. And so L-amino AADC is a very devastating disorder that very little movement and interaction with their environments. And this patient did receive the experimental viral vector, and then 13 months later, go ahead and play the right one. See a dramatic improvement in movement and interaction and just overall quality of life. So not a complete cure, but certainly a promising treatment for something that didn't have any real treatments before it. Okay, next slide. And so this study here with spinal muscular atrophy, this is really probably what turned gene therapy into one of the hottest, if not the hottest topics in medicine right now. So but this study was published in 2017 by Brian Casper's group. It was published in the New England Journal of Medicine. It's the clinical trial for spinal muscular atrophy and gene therapy. So to put this in perspective, patients who have spinal muscular atrophy type 1, it's an extremely devastating disease. 96% of the patients die before their second birthday. And if they survive beyond that, they're on permanent respiratory assistance. And just the second bullet point here, it's really just get to the nuts and bolts of it. With this experimental gene therapy, when they received an intrathecal injection, all 15 patients which received it were event-free by 20 months of age. And so it's SMA type 1 is commonly referred to as floppy baby syndrome. These children, they never move except for that which is caused by gravity. And you can look this up, look at some of the videos from the experimental cohort. You can see there's one video of a 6-year-old girl who's just running around her doctor's office. And then she drops to the floor and challenges her doctor to a push-up contest. And so it's really profound how much it's worked. But next slide. And so does gene therapy make sense? Now I'm going to talk about another success story with hemophilia, which is really relevant to adult Refsum disease, and that the target was the liver, which is what we would try to do for adult Refsum disease. So one thing that they were considering is, did it make sense for hemophilia? And the answer was that it already cost patients over $250,000 per year, and it required IV infusions every 2 weeks. And so with gene therapy, they could get lifelong treatment with one IV dose. So obviously that's more convenient. And then to move this video again. Then BioMarin actually has developed this gene therapy drug. In their latest clinical trial, 6 out of 7 patients that receive the highest dose are bleed-free, while all 7 no longer need prophylactic drugs. So that a tremendous success story. It's very relevant to potential treatment for adult Refsum disease. Go ahead, next slide. So the question which I was brought in here to talk about, will gene therapy be successful for ARD? I'm not psychic, but I am able to be able to look at this rationally. So things to consider for just preliminary analysis of will it be successful. First off is the size of the gene. Second is the accessibility of the system, which in our case will be the liver. Third, is there an appropriate vector for the cell target, for the hepatocytes, and for the immune response? Next slide. So with adeno-associated virus, there's a hard cutoff of about 4.6 kilobase pairs for the viral DNA. And so this is historically it was packaged single-stranded. And in 2001, there was a study which showed that if you use a mutant inverted terminal repeat, which essentially turned this single-stranded DNA into double-stranded or self-complementary, you had about a 10 to 100-fold increase in the efficiency of transduction. The trade-off with that is that the size of the DNA construct was cut in half to about 2.3 kbs. And I should point out that this 2.3 and 4.6 kb, that includes the gene as well as promoter regions and regulatory elements and poly-A tail, the whole everything. And go ahead, next slide. And so the relevance for adult Refsum disease, the gene which we are trying to replace, the PHYH gene, the protein itself is only 338 amino acids, or just about 1,000 nucleotides. And so it fits well within the constraints of self-complementary AAV technology. And so go ahead, next slide. That means for gene size, that is a definite check mark. So yes, we do meet that. Now talking about the accessibility of the system, can we actually reach the liver? Go ahead, next slide. And so like I was mentioning before, there are very promising results using AAV8 for the treatment of hemophilia A and hemophilia B. And so with gene therapy, one of the lucky things about using it as a platform for the treatment of diseases is that 90 to 95% of the process is identical, regardless of the disease you're looking at. And so the fact that all the uncertainties have been addressed with hemophilia trials, it suggests that this is obviously this would work very well for the treatment of adult Refsum disease. So prior data suggests that with AAV8 it would be relatively straightforward to reach the liver. And in fact, one thing that I could talk about with some of the scientists later on is how specific do we want to be for the liver. So AAV8, yes, it does reach the liver, but it also reaches several other organs. And so if we want it actually to be liver specific, then there are liver specific promoters that we could put into the DNA construct. And so that's just something that we can talk about as we're developing this project. Next slide. And so when you're talking about the target system, you need to take into consideration the route of administration. Some routes are easier for reaching certain systems. And when we're targeting the liver, the best way to reach it is through an intravenous injection, systemic and intravenous injection. There's also a talk about doing some retinal gene therapy for adult Refsum disease, to treat the progressive blindness that affects some individuals. But basically for systemic administration, we would go for intravenous. Next slide. So second point there, the accessibility of the system, that is a definite check mark. Yes, we can, prior studies have shown that we can reach the liver and transduce hepatocytes. And so the third thing, is there an appropriate vector for the cell targets? Go ahead, next slide. I'm going to start off by I'm going to talk about adeno-associated virus. And so this was first characterized in 1965. There's an old-timey photo on the right. The small little circles there, that is actually adeno-associated virus. It is single-stranded DNA human parvovirus. And importantly, it's non-pathogenic. It does not actually cause any disease in and of itself. It requires a helper virus for a lytic infection, and it's able to transduce non-dividing cells and confer long-term transgene expression. As I mentioned before, the single-stranded version, you can package up to 4.6 kb of transgene, and it's in place of the viral rep encapsulation. So rep is normally used for the life cycle of the virus, so the replication. When you're engineering these viruses, you want to make sure that they're replication deficient. And then the cap genes, which are responsible for capsid proteins. And there are dozens of naturally occurring serotypes, which offer different tissue tropism. As I mentioned before, as of December of last year, there are 250 clinical trials which use AAV and has a safe track record. And in 2012, the first gene therapy product received full regulatory approval in Europe. And in 2017, the US approved its first AAV products. Go ahead, next slide. So different AAV serotypes confer different tropism to different systems. However, they're not necessarily specific just to that system. For example, AAV 7, 8, 9 have been shown that they can transduce liver to hepatocytes, and AAV 8 being the one that worked very well for hemophilia. That being said, AAV 8, it actually can transduce multiple different systems. And so something to consider is that yes, you will get widespread distribution of AAV throughout the body, but just with no point to really reinvent the wheel in this case. So it worked for hemophilia, so we would use AAV8 for the treatment of adult Refsum disease. Next slide. So how to make a recombinant AAVs? So there's the engineering of this. It's a triple transfection process. You have your transgene with the ITRs, and you have rep and cap genes, which are responsible for making the capsid, making it non-replicative, and the capsid proteins. And then you also have add helper genes. You put it into a producer cell, and it produces the virus, which has the DNA construct, which you're trying to transduce into cells. Next slide. Next slide. Okay. Go ahead and open up the whole thing. There you go. That's enough. So the genome design, you have your inverted terminal repeats. Then you need to decide on enhancer or promoter, which you at the beginning. And like I said before, you can, for enhancer, for the promoter, you can decide, like, we just want to transduce as many cells as possible. And so you use a general high efficiency promoter. Alternatively, you could say you want to do specific cell type, in which case you do more specific promoter. Then you have your gene or coding sequence. For this case, it would be the PHYH, codon optimized cDNA. And you have the option of additional regulatory elements, which could actually boost expression, and finally the poly-A tail. And so for self-complementary AAV technology, which is what we would use, you would have a mutant ITR at the end, which would turn this into self-complementary or double-stranded DNA. Okay, go ahead, next slide.
Hey, Ryan, just to stop you for a second, I think we've had some folks say that there's an echo in your audio, and just wondering if you have a headset you could move to.
Unfortunately, I don't.
Okay.
If I speak slow, let me know if this continues. I'll try to adjust where I'm sitting.
Okay. All right. Sounds good. Thank you.
Open my doors. Maybe that will help. So, is that a little bit better? Go ahead and type if you hear me better now.
Yeah, it's fine on my end, but I think there might be a couple with, I'll put myself on mute too. Maybe there's some sort of echo from my side too, so.
That's possible. Okay, and then moving on to the immune response. Go ahead, next slide. And so it would be irresponsible if we didn't talk about some of the possible negative reactions to gene therapy. If you inject these viral vectors into a human, you will get an acute inflammatory response to the capsid. And it's also important to point out that 30% of all adults have an innate immunity to AAV. So if there was prior exposure in your life to AAV, then you will have an innate immunity. Now, that being said, with the ongoing clinical trials, it has been shown that giving steroids, anti-inflammatory steroids, prophylactically can drastically reduce the acute and the innate immune response to AAV. So this is something that is basically a potential problem with any gene therapy that goes on. And so yes, it's there, and there are ways to control it, but it's definitely something that needs to be considered on a patient-to-patient basis. And also for overexpression of gene protein, that doesn't occur very often, but it is something to consider. So is the transduction too good? Are we getting too much PHYH protein now? And if that were the case, we would just basically go back to the design of the viral DNA, the DNA construct, and do something to reduce the amount of expression of the protein. Okay, next slide. So in the end, all 4 boxes are checked off, and go ahead, next slide. So the answer to this question, will gene therapy be successful for ARD? And from the knowledge that I have, this is as close to a winning scenario as there can be. And so I would highly encourage this to be moved forward as best as we can. Go ahead, next slide. So some future work that hopefully maybe next year, if the world will allow us to meet in person, we may have some data on this. And so the first thing I would like to do in my lab is to develop the viral vectors. And we already have ordered codon optimized cDNA, and we will begin cloning that into existing DNA backbone, existing viral backbones. There is, and thankfully, there is an existing mouse model for adult Refsum disease. And so we would test the experimental viral vectors in the mouse model, test for efficacy. And then we could also do it like within a reasonable amount of time, we could do a pilot non-GLP safety study in rats. So go ahead, next slide. So things to consider for these translational studies. Is it safe? Is it practical? Does it work? Does it work the same in a mouse as it does in a human? Can I make enough to go into a human? How will I administer it? And can I do this in a human? So a lot of these have already been answered with previous studies. And some of the other questions, we can only answer those empirically. We won't know an answer until we actually try it. Okay, go ahead, next slide. So some additional considerations. At all times, there are labs around the world which are trying to develop better vectors. And so it's kind of like buying a new car or buying a new computer. How long do you wait to see where the technology leads you? And in a year, there could be a revolutionary new viral vector. Is it worth it to wait for that? And I would say for the prospects of treating adult Refsum disease, there's really no point to wait. What we have right now is basically off-the-shelf technology that's ready to go. And I wouldn't expect anything to come along to really be better than what AAV8 self-complementary technology can do. So in terms of additional considerations, I don't really think there's a need to to wait for the development of a better viral vector. But we also have to consider costs. So go ahead, next slide. So this was the slide at the very beginning, the very simplified way gene therapy works. You identify the gene, you package a working copy into a virus, and then deliver that virus into a patient. Go ahead, next slide. Next slide. In reality, it's a little bit more complicated. Yes, there are those 3 steps. One of the things you do have to do is you have to test the efficacy of the experimental viral vector in rodents. Okay, next one. And if that works, then you talk to the FDA, show them your results, and then you move on. Go ahead. To safety studies, and finally, if that works, if it's safe, you get permission from the alphabet soup of regulatory agencies for the development of a new drug. And go ahead, next slide. Or sorry, next. Yes, at each stage of this, you need funding. And so money is really what makes this work. So then, go ahead, next slide. How much does a new drug cost? So Brian Casper, the scientist who led the SMA trials, quoted it as his industry costs $1 billion capital investment to bring his Zolgensma drug to the market. That there's a little bit of give and take on that, so there may be ways to be more efficient with the funds. That was to develop the entire agency for making that. But for one good example here is for giant axonal neuropathy. This is a study which is being done by collaborators of mine at UT Southwestern. Over 6 years, they brought it just from the concept phase all the way to phase 1 clinical trial. And that cost about $3.5 million to get it to the point that it is. Now, that might be a bit of a sticker shock to a lot of you, but point to point out that we have several existing industry partners who are very eager to add disease indications to their portfolio. And in addition, proof of concept data with foundation support would go a long way to establishing new partners. And so you show that it works so in your mouse, you show that it's safe, and then there most likely will be an industry partner who would be willing to take this on. And of course, in addition to that, we could be applying for government grants and things like that. Go ahead, next slide. And so just to brag a little bit about the environment that I'm in right now, UT Southwestern is scheduled to be one of the only places in the world that they can actually bring it from bring gene therapy from the concept phase all the way to the clinical trial. And so scheduled on September 14th, they are scheduled to make their first 500-liter GMP viral vectors. And so just to keep that in perspective, that's fairly rare. And I just wanted to brag about the environment. Okay, next slide. And so at UT Southwestern, there's a huge pipeline of diseases which we are looking at. And some are in the discovery phase, some are in the IND or investigational new drug enabling phase, and some are in the clinical trial. I already mentioned GAN. We also have Batten disease clinical trial going on. Now, 2 of these diseases, which I'm currently working on, Angelman Syndrome and Peroxisome Biogenesis Disorders. And obviously, adult Refsum disease is a peroxisome disorder, so that just to show you that is what other things are being going on right now. Okay, next slide. So that's the end of my talk. I want to thank all the collaborators I have around the world. First off, I would like to thank the members of my team, Hairi Kang and Ayman Rahim, and also Steve Gray, who's really been a faculty mentor for me as I've developed my gene therapy arm of my lab. And also, I'd be remiss if I didn't thank Joe Hasia, who basically is the reason why I know Christy and have been introduced to the Global DARE Foundation. And next slide. And I also want to give a shout out to a whole bunch of new collaborators. We had our first Zoom meeting this past week just to talk about ideas and ways forward for producing grants and things like that. And so, yeah, we'll be having regular meetings to discuss future projects. And last but not least, I want to thank Christy DeMarco. You're doing everything right so far, Christy. So one of the biggest indicators of whether these things work is the engagement of patient and family advocacy groups. And so keep it up. You're doing everything right so far. And that's all I have. Next slide. Next slide. Okay. Now I am open to taking any questions you may have. Thank you very much.
All right. Thanks, Ryan. We do have quite a few questions rolling in. I'm going to actually stop sharing the screen so they can see us in nice and big format. We'll be right big on the screen. So I'll start with questions in the Q&A box within Zoom. As a reminder, for those of you who are on the phone, you can press *9 on your phone to raise your hand to ask a question as well. So the first question, is gene therapy very expensive treatment? How much may it cost approximately in general? I know you touched a little bit on that already, Ryan, but I don't know if you want to expand upon that in any more detail.
So yes, it is expensive. If you heard about the SMA drug, just reading regular websites or things like that, the headline normally said, most expensive drug ever produced. Yes, that is true. One thing to bear in mind, though, is that it is a one-time treatment. At least to the best of our knowledge, it's a one-time treatment. Ask me in 50 years and I may revisit that. But as of right now, it is expensive. It is expensive to produce, but what you're looking at is one-time treatment for a drastic improvement in quality of life. So yes, sticker shock is something to keep in mind. But
I know when I attend to some of the conferences where they talk about gene therapy, they're really starting to focus in on the production side of things, right? Where they could potentially reduce the cost for production, so hopefully in the future we'll see it come down, right? Which would be great. So if gene therapy was successful in adult Refsum disease, in what areas of the disease could we hope to see improvement?
So that's I will say that when it comes to these collaborations, one downside of it is that my knowledge of adult Refsum disease is actually rather superficial. I know the gene that's involved and I know some of the manifestations of the disease, but we are targeting the liver, and so circulating Phytanic acid levels should go up. I would, maybe even Christy, you would know more than I would. What is affected first by the circulating Phytanic acid levels? I would say that the first thing that would be the first improvements you'd see are the things that go the first, but I could be wrong about that. So yeah,
I think the thought is, and I know, as we talk through in the research group, that there's more research that needs to be done. But I think, one of the things we see when we increase our Phytanic acid levels, that we see the neuropathy and the ataxia symptoms, the skin issues. Those are the things that, pop first for, as it starts to rise. I think the question has come up around, would it actually reach the eye? If we fix it in the liver, would it actually reach the eye? And, is the Phytanic acid, because the retina has its own metabolism, would actually you be able to affect that? And would you actually need gene therapy both for the liver and for the eye? And so I think those are some of the things that we would look to research when we would put forward those preclinical studies, right, that we've been talking about in our research group. What are the studies that we can do to help understand what are the best approaches? But we don't know for sure, what would be exactly improved upon, but that's the whole reason for the preclinical study, to see whether it would be effective or not. So
yes, indeed, I would say that at the very least you would see a slowing or a stopping of the progression of the disease, whether you would see a correction of where the patient is currently at. you could be hopeful that that would be the case. But what I would really want is that the disease is slowed and that's reduced.
And they are doing, recently they were doing some studies relative to the PEX1 gene, right? And they were inducing fixing it in the liver, I believe, and did see some positive impacts in broader areas than they thought. So I think, I think it's optimistic of how that research might translate to what we might see so I think your research along peroxisomal disorders altogether, right, would provide good insight into how things might be helpful from a adult Refsum disease perspective. So, all right. So the next question, hopefully I'll say this right. If prophylactic steroids are required, is this typically a lifetime requirement or needed for a period of time to overcome the body's initial response?
It's just an initial response to the viral vector. So the viral vector will be in the bloodstream for several weeks afterwards, but then after transduction of the viral DNA, then it is eliminated from the body. So you will not need lifetime steroids to manage an immune response. Okay,
great. So I think you just answered this too, but just to make sure, so does this mean you will have to have drugs for immune system rejects of treatment for life? I'm assuming the answer to that's no, but.
I mean, it will be, there are instances of a severe immune reaction. This is the most common question, and rightfully so when it comes to gene therapy, because it is a potential downfall for it. It is what, for people who are not, or for most commonly, people are referred to the Jesse Gelsinger case. That, I should point out, was with adenovirus and not adeno-associated virus. But yeah, so that it is something that the doctors, after they inject the virus, will have to manage and keep control of. There's also some studies ongoing right now which are investigating whether we could incorporate a small DNA construct which would be basically anti-toll-like receptor, that sort of thing, which may reduce the innate immunity to that 30% of the adult population which has that immunity. So yeah, I mean, these things need to be taken into consideration, but from what we've seen so far, prophylactic steroid treatment should be effective for most patients who receive these viral vectors.
Okay, great. So I think you've answered this one, but let's just make sure. To what extent does gene therapy reverse the impact of adult Refsum, in particular gradual loss of eyesight?
So yeah, I did mention this earlier. At the very least, what I'm hoping is a slowing or halting of the progression of the disease. When it came to the inherited blindness study, there was actually a correction. Whether or not we'd see that, we really don't know until we do it. We can hold out hope that the deficits which have accumulated over the years in adult Refsum patients, that could be corrected. That would be ideal. I won't say that will definitely happen, but what we would be hoping for is the halting of the disease.
Yeah. You would think with the lessened circulating phytanic acid, if you were to correct it in the liver, and we would have much less possibility of circulating phytanic acid, it probably won't necessarily reverse, dead retinal cells, but it would potentially halt the further degradation. But until you actually do the research, you don't know for sure. So. And the mouse studies would probably provide insight into that as well. So, okay, the next question here, it says, typically we have got the position of, typically when you've gotten to the position of using gene therapy on humans, how long would a non-US based patient be expected to wait to access this?
Well, a non-US based patient, that's a little bit more difficult for me to answer because I'm not as familiar with the European regulatory agencies. I will say that Europe was first to approve, so maybe there are ways for it to go faster. In the United States, there's compassionate clinical trials out there, so that you could actually get it earlier if you are willing to basically absolve responsibility if something goes wrong. And also, it's when I see the timeline, it's actually the FDA wants to see these get to the market as fast as possible. They recognize that this is possibly as revolutionary in medicine as we're going to see in our lifetimes. And so there is a strong push to make this go faster. And with the GAN trial, the giant axonal neuropathy, that took about 5 years to go from the concept phase to the clinical trial. And that was basically just with government grants. And so if you were to have an industry partner that has existing capital ready to spend, you could probably cut down the time on that as well. But in terms of actually getting all the regulatory agencies on board in Europe, I can't really answer that question, as I'm not as familiar with European regulatory agencies as US.
Yeah, okay. Well, thanks there. So is there an optimal age for the best results?
Newborn, but yeah, typically with gene therapy trials, at least the earlier you catch it, the better. I wouldn't say anybody should exclude themselves if they're older. I mean, obviously this is called adult Refsum disease for a reason, but if you want the best effect, you would want the earliest diagnosis possible and to start it as early as possible. It's probably, I mean, Christy and I were just talking before this, and the manifestations of her disease struck her a lot later than some of the other adult Refsum patients. And so there's probably not a defined age that you would want to start this, just broadly speaking, as early as possible. Like I say, we're looking to halt the progression. And if there's a correction of existing deficits, and that would be a plus, but that's how I would answer that, as early as possible.
Yeah, and I think we've been in discussions around, could we introduce newborn screening for adult Refsum disease? Because obviously the default gene exists, from birth, right? And so could you introduce newborn screening, or, as genetic testing becomes more available at the point of birth, I think you would want to introduce it as soon as possible, because obviously you start to eat very shortly after birth, right, and start to accumulate that phytanic acid, since you're not able to process it. Even though some of us might be later in our presentation of it, it's still there, right? And so the sooner we could, it'd be great to have no symptoms ever, right? So the sooner the better. All right, so let's see. So I don't know if you can answer this question, Ryan, but the question is, have all the faulty Refsum genes been identified? And I think it's probably not a question.
Well, the one that we would be working on initially is the PHYH gene. Also, PEC7 is also, I believe, could be packaged self-complementary. So I don't think there's anything beyond PHYH and PEC7 that have been identified, but please correct me if I'm wrong on that. There are labs that I know of, a lab in Caltech which is looking at gene therapy for PEC7 patients. And so we would start off with PHYH because of that. If there's anything beyond that, I'm not entirely sure.
Yeah, I think in Anthony Wierzbicki's presentation, he talked about a couple of others, but really 90%, I think he said, of the Refsum patients that have been identified to date, it's the PHYH gene. So that is obviously the most prevalent gene as it relates to adult Refsum disease. So, okay, I think we have a couple other questions. They're not necessarily Refsum related, but more from a gene therapy research perspective. Is there gene therapy research related to the treatment of sickle cell anemia?
Yes, there is. I don't have many details that I can pull off the top of my head, but yes, there is. And in fact, that was one of the first ones that they tried to go for. I'm trying to, can't remember the gene they were trying to correct with that at the moment, but yes, there are for sickle cell anemia.
Yeah. I think the other question was whether it'd be useful in the treatment of cancer.
Oh, yes. So you may, it's not really gene therapy so much as cell therapy. So CAR T therapy is big in the field right now. Yes.
Yeah.
So this is sort of a personal thing for me because my mother is actually suffers from stage 4 breast cancer right now, and I have been looking into this, not professionally, but sort of personally, trying to see what's the latest out there. So yeah, the CAR T therapy has been shown to work wonders for leukemia. In terms of, it depends on the actual type of cancer you're talking about. So, solid tumors is really the next, as I understand it, is the next thing they're trying to go for CAR T therapy.
Yeah. Okay. Another question. So, obviously we talked about the benefits of early introduction of gene therapy, as early as newborn. But what would be the benefits of doing it later in life, say at age 70? Would there be really minimal results and not, as much of a benefit later in life to do that? I have some thoughts, but I'll let you start too.
Age 70. Well, I mean, I think there probably would be a benefit no matter what age. Like I said, if it's getting worse, you want to help. The first thing you want to do, and if you are diagnosed with the disease, is you want to halt the progression of it. And so at any age, I would say it would be important to start. Whether 70 years old, I'm not going to surmise to know the answer to what the disease looks like in a 70-year-old patient. But I would say no matter what age you're at, you should try and get treated. Yeah.
I would think too, this is just me talking, is that, I think one of the things that impacts our quality of life significantly beyond, say, vision loss and hearing loss and things is our ability to, the diet, is obviously a challenge, and that you have to be very restrictive and very disciplined about your diet. And if you were able to fix the gene in the liver, and it allowed us to be able to actually consume more Phytanic acid, that would be, obviously a benefit to our quality of life, and that it would open up more foods for us. Hopefully I live to the age of 100, if I was 70, that gives me 30 more years to be able to enjoy more foods, right? So I don't know. I think there could be lots of benefits to, doing it at any age. And hopefully, as time goes on, the cost will go down as well, right? So another question, and I think this is maybe a little bit, it would be good to educate a little bit on what's the difference between what we're proposing here versus, say, Crispr, and what they're doing around, gene editing.
Yeah, so gene editing, that is also an avenue to explore. When it comes to CRISPR, I would prefer for this, if I were king for the day, I would prefer for the field to use that more for genes which are too large for gene replacement therapy. There are some off-target effects of CRISPR which they're only beginning to investigate. And for something like Duchenne muscular dystrophy, which Eric Olson, a collaborator here at UT Southwestern, is looking at. They started a company for using CRISPR-based technology for gene editing for that disease. And in that case, dystrophin is actually the largest gene in the human genome, and they're using it to correct that, and I've seen very promising results. Whether or not there's a need, whether that would be better for the treatment of adult Refsum disease, it probably would be relatively similar if the technology got to the point that we're at right now with just straight gene augmentation. But as of right now, I wouldn't even say I'm biased towards gene augmentation, because I would really like to see where gene editing goes. But at the moment, I wouldn't say there probably would be any tangible increased benefit to doing CRISPR-based editing versus gene augmentation for a gene as small as PHYH.
Okay, great. Can you comment any on the difference between gene therapy and, say, stem cell therapy?
No, not really. And so, no, I honestly, I'm not going to pretend to know much about how stem cell therapy would help in this instance. We're trying to correct the gene in this instance, and maybe for stem cell therapy, if there was like a loss of tissue or something like that, and you wanted to regrow it. But like I said, my knowledge on that is rather superficial. So I'm not going to comment much on the benefits of stem cell therapy in this instance.
Yeah, I have, in a little bit of discussions with our researchers, have talked a little bit about, stem cell therapy and the potential of the correction for the retina. And because once a retinal cell is dead, gene therapy is not going to help on that front, from what I understand, but that there is the potential of something like stem cell therapy actually regenerating the retina cells. And so I think that's, now it's probably much further off as a possibility, versus gene therapy is something that's already, moving along, and something that is more of a viable option in the near future, where I think what I've heard about stem cell therapy is that's a little bit further off as far as a possibility for us. But I think we're in this for the long haul. It's not everything's gonna happen overnight. And so, we'll continue to look at, what are the viable treatments for Refsum as a whole. So, all right, I think that is the last question that we have. Let me just double check and see if there were any hands raised. I don't see any. So I think that's it. So, I want to thank you, Ryan, for coming today and talking with us about gene therapy as a potential future therapy for Refsum. It would be exciting. I know we've got a lot of very interested medical and scientific advisors, some of which you showed up on the screen, that you're starting to collaborate with, and we're starting to meet with regularly. Our hope is to, apply for an NIH grant to start some of this preclinical work, and also, hoping that the foundation can actually support through its fundraising efforts some smaller ventures. Little pieces of information that we can provide into this is helpful. The patient registry will also be very helpful. I know, especially in getting industry engagement. Ryan and I have had many discussions around, if we can increase our number of patients, get folks diagnosed earlier, have more people connect with us, have more people register within our registry, it'll make adult Refsum disease a much more viable, candidate for an industry partner to be interested in funding this for us. And so very excited to see how that goes. And so thank you. And thanks everyone for for attending. Today, that ends today's webinar, and have a wonderful day.
Okay.
Thank you, everyone.
Thanks, Chrissy.